Hello everyone, so we're going to continue exercise 10, which is kingdoms bacteria, protista, and fungi. So I already covered kingdoms bacteria. Now I'm going to move on to kingdoms protista. So let me go ahead and share my screen here. Okay, so just like a quick little recap, we are studying the different kingdoms that make up life here on Earth, and we've already talked about our prokaryotic kingdoms. Those prokaryotic kingdoms are Kingdom Archaea, which I briefly mentioned, and Kingdom Bacteria, which we spent some time discussing. We discussed how we can classify bacteria based on shape, based on color, depending how they react to the crystal violet gram stain. We also talked about how you can grow them on agar plates and the different characteristics you can see on an agar plate. We talked about how both archaea and bacteria can be both beneficial as well as detrimental. And now we're going to move on to the eukaryotes. So in the eukaryotes, we have the kingdom Protista, fungi, Plantae, and Animalia. Today we're going to for now, we're going to cover the kingdoms Protista and fungi. Next week, we're going to discuss plants. And then the following week, we will discuss animals. So your final lab practical will have a lot to do with kingdoms. Obviously, it's going to have material from the other exercises as well. So exercise 8, gene expression and DNA technology, as well as exercise 9, evolution and natural selection. And then it's going to have all the kingdoms on there. When you're working on your lab practical, a lot of it is going to be identification. So you will be given an image and you will have to identify which kingdom it belongs to. You will also have to identify some key characteristics of that specific species you're observing. So when you think about the kingdom Protista, remember, although these look very, I guess, ancient or very simple, these are still eukaryotic organisms. They have membrane mound organelles like a nucleus. And remember, these protista are going to look very different. We have the animal-like protista, the fungal-like protista, and the plant-like protista. However, they are not animals, they are not plants, and they are not fungi. This is a very diverse kingdom. And as we discussed earlier, it is a catch-all category. So if it does not fall into the animal, fungi, or plant category, it automatically gets clumped into this protista. So it's a catch-all category. It's a very diverse kingdom when it comes to whether it is a unicellular organism or a multicellular organism. So a unicellular organism would be, for example, amoebas, which I will show you in the next slide. We also have some multicellular organisms. And then as we go through these organisms from the kingdom Protista, I want you to think, what is its, is it a unicellular or multicellular organism? 1. 2, how does it, what is the mode of nutrition? Is it an autotroph? So, does it survive through photosynthesis? Or is it a heterotroph? Does it consume other heterotrophs or autotrophs? Or is it both? Some species can be both. Some species are both, should be up here, I'm sorry. It's not under types. And then once again, there's different types of protista: fungal-like, animal-like, and plant-like. When I say some species are both, it means they could be both heterotroph or autotroph. Remember, these are not, they're just animal-like, they're not actual animals, or they're not part of the kingdom Animalia, or they're not part of the kingdom Plantae. So as we go through the kingdom Protista, this is what you're going to do. And the only way you are going to be successful on the lab practical is if you actually take the time to do these exercises in your lab manual. So in your lab manual for activity 5 and activity 6, you will sketch the specimens that you are presented with. So for example, you will sketch a preserved amoeba. You will also sketch a live amoeba, and I will show you which slides you will use to do this. And then you will answer the following questions. Where are they found? Are they free living or parasites? How did they obtain their food? Are they mobile? If they do move, what structures do they use to mobilize themselves? So are they using pseudopods? Are they using cilia? Are they using flagella? Like how is this organism able to mobilize itself? Are they unicellular or multicellular? And do they have any unique structures or properties? Is there anything unique about this organism that you can help classify it or help identify it? So we're going to start with animal-like Proteus, and we're going to look at amoebas. We're going to look at a type of ciliate, which is a paramecium, and then different types of organisms. So for example, the trypanosoma, which is a blood parasite, which is shown here. So these are the images you will, these are the prepared slides. So where it tells you to look at preserved amoeba, preserved trypanosomes, etc., you are going to sketch these images shown here. I also have provided you with some information about these organisms. I adapted this from your guys' lecture instructor, Dr. Scott. So for example, for amoeba, you would answer where are they found. They can be found in freshwater environments or in the soil. Are they free living or parasites? They are typically free living, but they can be pathogenic, so they could also be parasites. How do they obtain their food? They eat bacteria, fungi, and decaying organisms. Are they mobile? What do they use to mobilize themselves? They move by means of cytoplasmic streaming to create pseudopods. Are they unicellular or multicellular? They are unicellular. Do they have unique structures or properties? And they have contractile vacuoles which expel excess fluid from the cell. So you will go through, and then here are the amoebas in motion. You'll play these videos to draw the live amoeba. And then this is an amoeba eating a paramecium. So you could play those videos there. And then this is the paramecium under the microscope. This is going to be the mobile one. And here's some information of paramecium, so you can go ahead and answer these questions. These are going to have cilia all around them. And they are classified as ciliates because they are covered with cilia. You want to know how they use it, what the cilia is used for. And then you're just going to move on to this organism here. And here's some information about it. Then you're going to finish off the animal-like Proteus with trypanosomes. So these are free living in water, both fresh and salt water. These are mostly parasites. They are parasites found in the blood, as you can see with these red blood cells here. These are unicellular organisms. They have the flagella, which you can see here, as well as here to move around. And in our lab, we actually do work with trypanosomes. So we are given slides from the state, and they are smeared with patient's blood. And we have to identify the species of trypanosomes that we see in the microscope slide. And the way you make these identifications is using clinical data. So has this individual traveled anywhere? For example, did they go to West Africa? Did they go to different regions of Africa or other countries where they could have been exposed to these parasites? So you use clinical information, but you also are going to look at not only the structure of the parasite, but also the red blood cells. You're going to see, look for stippling of cells. So the stippling, you'll look at the stippling to make your determination of which type of trypanosome you are looking at. So you have to actually identify the type of trypanosome you are observing under the microscope. So, but here we're just looking at them vaguely. You don't have to know all the subspecies of them. So don't worry about that. I just kind of like giving you guys information on how we actually use this in the lab every day. This is, we don't really come across this in our patients in San Bernardino County. However, we do have to go undergo state testing. So our lab receives different tests from whether it's the government, the state government, or the national government. So we get tests from, for example, like the CDC. We get tests from commercial labs and agencies to test to determine our competency on various subjects. So things we don't usually deal with on the daily would be, for example, trypanosomes, botulism, Bacillus anthracis, ricin. However, we do receive these specimens in the lab and we have to test them just to make sure that if we ever did encounter one of these samples, we would be able to identify it and to successfully do the analysis. So then you're going to move on to plant-like proteus, and you're going to do the exact same thing. You're going to draw these specimens or these organisms in your lab manual, and you're also going to answer the questions that I presented in the beginning of this. So we'll go through this for all the information is here for you. And these are diatoms, so these are very beautiful under the microscope. If you watch One Strange Rock with Will Smith on Netflix, you may have learned about diatoms. The other really neat thing under a microscope, this is going on a tangent, is urine. If you were to look at urine under the microscope, like a urine midstream under the microscope, you would see like these beautiful crystals. It reminds me a lot about diatoms whenever we do urine analysis. But that's just a side note. So you will go through this for the plant-like proteus, like I said, and complete these activities. The best way to be successful on the last lab practical would be taking the time to actually complete the activities in your lab manual. If you have any questions, please let me know.